Hello, I'm Heba Sailem. I'm a research fellow at the University of Oxford, and I will talk today about a machine learning framework for analyzing large-scale genetic screens. Genetic screens offer a powerful tool to study gene functions. And the idea that we can perturb genes, for example, knocking them down, and observe how the phenotype of the cell change. And this can give us indication on what the gene is doing. So, for example, here we have cells that are stained for DAPI and rotavirus infection marked in green. If we knock gene like gene 1 here, and we observe that the cells have increased roundness, we can infer that the gene has a role in contractility. We knock another gene and we observe that the cells become very huge compared to control cells, then we can infer that the gene might have a role in aging or senescence. If we do this for every single gene, then we can start linking genes that have similar phenotypes and say that they are functionally related. So the dataset I have been using is a genome-wide screen that have 18,000 gene knockdown. So here in a multi-well plate, every square will have a different gene knockdown. And we are imaging the entire well in the plate. So this is result in around 6,000 cells for every gene knockdown. And these cells are stained for, as I said, DAPI is in blue and green is rotavirus infection. So in total, the screen resulted in over than 500 million cells. This gives us a lot of information that we can use to learn not only about the phenotypes at the single cell level, but also at the different levels. For example, changes in the specific distribution or the arrangement of cells, etc. And to be able to harness the power of these datasets, the first thing is to use automated image analysis. So here we can use image analysis to segment every nucleus and every cell and extract various features such as size, shape, or texture. So other features we can extract is the context of the cell. So whether the cell at the edge of the colony or not, the local cell density, if it is in the middle of the colony, then it is higher density than if we're going toward the edge of the colony. And also we can train classifiers to identify various cellular states like apoptosis, mitosis, or infection. And the task after that is how we can link these genes to functions. So the traditional approach employ clustering. So we cluster the gene profiles, and here every gene knockdown will have a set of features to see which genes have a similar phenotype or a similar signature, and then we assume that they will have a similar function. So once we have the clustering, we can look what are the clusters and what are the functions that the clusters enriched for. Although this seems like logical, the challenge is that when we do that, it's very difficult to interpret what the cluster means and what the phenotype means. And we end up saying, okay, we don't understand these results. Let's focus on the features we understand. So in most studies, only 1% of the data would be analyzed. And this means that we're losing a lot of information on the gene functions that we can use to learn and advance our understanding of the gene role in the various phenotypes we have in these screens. So this is the challenge I wanted to solve. How we can systematically identify the relationship between genes, functions, and phenotypes. So the idea that I came up with is, instead of doing clustering, why we don't start from what we know. So if we know that certain genes play regulate a certain function, we can look in the data if these genes have a specific phenotype. If we find such a phenotype, then we can say, okay, what are the other genes that have a similar phenotype? And from that, we can link those genes to the same function. So because this approach is guided by our knowledge, I call it knowledge-driven machine learning. And to make it in a computational framework, this is basically how it looks like. Let's say we have a function like cell division, and we have a certain genes that we know that they regulate this function and along their depletion phenotypes or features. So then the question is, do these genes have distinct phenotype from a random set of genes? So we can look if there is features that can distinguish cell division genes from a random set of genes. And once we identify the subset of features, then we can train a classifier that separates the cell division genes from a random set of genes. And once we have the classifier, basically it is about applying it to other genes and seeing which ones will score positive. So here the orange indicate the positive ones, and the confidence in the prediction, because we can look at the phenotypic similarity. So where we will get this prior knowledge, I use gene ontology. So I selected gene ontology terms that have between 100 to 500 annotated genes, and I trained a classifier for every one of those. So this is how it's look like. We select a function like cell cycle, train a classifier to distinguish cell cycle genes, and then apply the classifier to see other genes that are phenotypically similar and rank them. Of course, given a certain function, we will not observe all the function in a specific cell type or in a specific context or based on a subset of markers, right? So in case the classifier doesn't result in certain accuracy, then we don't use it. And at the end, once we apply the method to all the selected functions, we will have multiple predictions for each gene and what are the prediction in their involvement in different functional terms and the confidence of these predictions. So one important thing with this approach, then this prediction would be context driven. So it's driven by the data. So whatever we are discovering from this data set might be specific to the cell line or the cell type or the specific context of the experiment. And that's why I called my method knowledge and context driven machine learning, or KCML. So now the question is, are KCML predictions any good? Can we use those and trust those? So I came up with a way to systematically validate these predictions. So while I was developing the technology, new annotations were added to the gene ontology database. So thousands of new annotations. So basically what I've done is to download these annotations and look if how many of the KCML predictions are now in the gene ontology database. And apparently there is a significant portion now actually in the gene ontology database confirming that KCML can predict novel gene functions. So in summary, what the main take-home message is that using this approach, the knowledge-driven approach, we are able to extract far more information from genetic screens than what is currently possible. At the end, I would like to thank the Wellcome Trust for awarding me the Sir Henry Wellcome Fellowship, which has been an amazing opportunity to work with different groups, and mainly my collaborator, Lucas Pelkmans at University of Zurich, who provided the colon cancer dataset and advised me on the work, and my research sponsor, Ian Richard at the University of Oxford. And thank you for listening, and I'm happy to take any questions.